Good morning, everyone, from Durban, South Africa. This is Serban Proches speaking to you on this 2020 Humboldt Day. I was quite excited to be asked to contribute because Humboldt is one of the guys I hold responsible for calling myself a biogeographer today. This is actually my Humboldt book that I grew up with back in Romania. And this is something that I definitely did not grow up with back in Romania. This is a dwarf chameleon. They're quite common here in Durban, although they're by no means easy to find. So today I'm going to speak to you about the global distribution of ancient plant and animal lineages. Before I do so, I want to give an opportunity to some of my colleagues and students to introduce themselves and to tell you a bit about the biogeography research happening here in Durban.
Serban Proches from sunny subtropical South Africa. I'm Sid Ramdani. I'm a botanist that is interested in taxonomy, evolutionary history, and biogeography. My interest in biogeography stems from the work of Frank White on Afromontane vegetation. There's no doubt that Frank was heavily influenced by the pioneer work of von Humboldt on mountain systems. My pet family is the family Asphodelaceae, or the Asphodeline aloe family. This family has a huge center of richness and diversity in southern Africa. The genus that I'm most interested in is a genus called Niekerk, or red-hot polkas. Niekerk has a strong presence in Afromontane grasslands across the African continent. I work very closely with Serban Proches on a number of projects, but mostly projects that deal with regionalizations at multiple scales using different lineages. Take care, be safe, and hope to see you in South Africa soon.
Hello, let me take this occasion to introduce myself. I am Erwin Sieben, and I'm working as a biogeographer at the School of Agricultural, Earth and Environmental Sciences at the University of KwaZulu-Natal in Durban. So yeah, my main interest is in wetlands, also in forests and montane ecosystems. And want to also introduce you a bit to the bioregion where I live and where I do most of my work, which is the Pondoland-Tongaland region on the east coast of South Africa. So the east coast of South Africa is very distinct from most other parts of South Africa, in that southern Africa is a very arid part of the continent. The east coast of South Africa, as you can see around me here, is much more lush, and regularly you can find forest patches. Basically the vegetation around here is a mosaic between forest, savanna and grasslands. So some of these forests have interesting elements. The coastal forests around Durban, what you find very commonly is, for example, this plant here, Strelitzia nicolai. This is a very small family which mostly occurs in South America. It's unique in South Africa and also with some species in Madagascar. It's a very common species around here, but biogeographically it's quite a unique element within the African continent.
Greetings to all. My name is Bonius Omjaja. I'm a master's student at UKZN. My research is looking at the global biogeography of recalcitrant seeded taxa.
Hi, I'm Ashna Bidiakshi. My postgrad research at honors, masters and doctoral levels all focused on Durban as a case study. My research included projects which focused on bush encroachment in grasslands, mapping the distribution of ancient endemic lineages, as well as studying biological invasions in an urban context.
Hello, my name is Michael Cheek. My PhD is looking at arboreta in South Africa, their status and role as sources of woody plant invasions.
Hi everyone, my name is Lindan Mavimbela from Eswatini. In my PhD studies, we studied invasion by the different growth forms of trees, vines, and shrubs. We looked at the dynamics in invasion, looked at how canopy cover influences invasion. We looked at niche spaces. We studied the coastal forest of eThekwini municipality in Durban, South Africa. Thank you.
Hello everyone, I'm Loki Snyman. I'm the curator of entomology at the Durban Natural Science Museum. Behind me is our collection room housing around 250,000 insects, mostly butterflies. I, however, is more interested in hematophagous dipterans, biting flies, mosquitoes, and horseflies. Then I have a special love for the biology and systematics of neuropterans or lacewings.
So having done the introductions, I can now move to today's presentation. It's a presentation about new ways of mapping biodiversity and new ways of perhaps understanding biodiversity. Much of it is about mapping ancient lineages on a global scale, and much of it is also things that myself, Sid, and Erwin have been discussing over the years, and some of it has materialized as articles. So I'm hoping to get your mind off the COVID crisis for a bit and maybe take it back to a crisis that's been around for much longer, the biodiversity crisis. I don't need to persuade you there is a biodiversity crisis. Every year and perhaps every month there's another shocking twist as to how much of a crisis it is. I'm personally still stuck on what shocked me most in 2018, which has to do with biomass distribution, and more precisely, mammalian biomass. It turns out, for those that haven't seen this one yet, that we humans now weigh 9 or so times more than all the wild mammals on Earth put together. On the other hand, the mammals that we keep for our food substantially weigh more than we do. So just a shocking thought for the day to start with. Also beautifully and clearly presented data and very straightforward for everyone to understand. I'm saying this because my feeling is, as biogeographers, we're not really getting our results across as broadly as we should. And maybe to improve on this, we need to reinvent a bit the way we understand biodiversity and the distribution of biodiversity. I think we need to work more with the social science guys, and what I'm showing you now is just a couple of studies that I found useful in understanding where we stand in terms of what people associate biodiversity with. To summarize this from my own end, how do we work with biodiversity? What are the key verbs in biodiversity research? Well, we sure measure biodiversity a lot and we map it with the aim of eventually conserving it. As a biogeographer, I may be tempted to overstate mapping, but we have to agree that mapping is essential to conservation. Before we even start measuring, though, we need to think, and we probably need to think a lot more. How exactly do we comprehend biodiversity? Historically, we have understood biodiversity in terms of species. This is understanding through naming, which is an individual-based approach, similar to the way we know people. I know you by your name, I know him by his name, and so on. It's important to stress that in terms of psychological processes, this is not the same as counting, which would be measuring proper. Even so, we have transferred the species approach to measuring, and to this day it remains the main way we measure and map biodiversity. More recently, we also have phylogenetic and functional approaches, and that's a great idea to diversify a bit. This is just one example of a recent study that maps all three: species diversity, phylogenetic diversity, and functional diversity in the same place. And it's nice to see that they're all quite well correlated. Of course, they're not perfect substitutes for one another. But just getting back to the basics of mapping biodiversity. We now have tons of data on species distributions, and the map you see at the top is the GBIF data the way they were a few years ago. There must be quite a bit more by now. And the bottom one is just a nice smoothed map showing the typical latitudinal gradient in species richness, something we know very well. In fact, one of the things we know best in biogeography. The question is, do we actually manage to convey this message to the broader public? So I'll give you a couple of seconds to take this slide in. Don't get me wrong, I'm not accusing Sir David of having actually said these words in the middle of the rainforest or in the Antarctic or in the Namib Desert. Just the sense that I'm left with after watching most wildlife documentaries is, okay, biodiversity is amazing, it makes a plan anywhere. And I'm feeling a bit bad talking down this approach. It's quite a romantic idea of biodiversity, and it's probably brought in tons of money for conservation. The question is, after having told the story for a few decades, maybe could it be possible that we can tell a more detailed story now? And to be able to answer that, we need to understand a bit more about our perception of biodiversity. We need to understand what in our understanding of biodiversity is perennial, cannot be changed, and what we can maybe start working on, taking in more, providing more detail. Here are just a couple of studies that can maybe give us some insights. This is a 2012 paper by Dalimer and his collaborators, and it's basically telling us people love to see greenery. And even when they say they love biodiversity, what they really love to see is greenery. Here is another one from 2014 by Schwartz and colleagues. And it tells us people do really poorly in terms of estimating biodiversity. This study and others are essentially painting a picture where we see people taking in maybe 10, maybe 20 species in any given group, let's say 20 birds or 10 or 20 plants, and any increase on top of that is not really digested any further. Now, if you look at it that way, rainforest is really the same as desert and almost the same as the Antarctic. So we have to ask ourselves, is it just the general public, or could it be that even ourselves, biogeographers, ecologists, researchers, whatever we call ourselves, are actually in the same boat? Of course, we have a better theoretical understanding, but we really let those numbers sink in. Do they make it to our subconscious? I'm tempted to say no, or at least not commonly. The estimate of how many species there are out there, it's in the millions, 10 to the power of 6, maybe 10 to the power of 7. So leaving aside the common objections about how species are being used in conservation or in biogeography, and I've had my own species rant published earlier this year, and you're welcome to read the whole thing. But leaving aside that type of objection, what does it mean to the human mind if we say the number of species out there is in the million? What does it mean except biodiversity is unfathomable? And saying biodiversity is unfathomable might have some sort of a PR value in an overwhelming sort of way, but I would prefer to be able to give people out there a slightly more detailed way of looking at biodiversity if they are to chip in for conservation. So what categories have we got that are more manageable? The most obvious one is higher taxa, genera, families, and so on, but they've been accused of being unnatural. More recent studies, like the work of Elise Humphreys that I'm showing you here, indicates that things are a lot more complicated, and species are not necessarily the most natural classification unit around. Besides that, the way people have been historically classifying living things, especially in traditional society, traditional societies, and I'm showing you here a book by Brent Berlin that's very relevant to this statement. The way people classified things does give a bit of weight to the single word units that are more or less equivalent to genera. So perhaps there is some potential in higher taxa. The trouble is, though, and I'm sorry to sound like a grandpa, Higher taxa aren't what they used to be. Genera with too many species are being split to make them more manageable. Families with too many genera are being split to make them more manageable. But the whole naturalness of higher taxa was that they were recognizable, ideally by one common trait, or if not, at least by a combination of traits that one could easily pick up. Let me just give one example, geckos. We used to know what a gecko was. All the geckos you see here, they've got something in common. Let's call it gecko-ness. They used to be one big happy family. Now, because they've been around for a long geological time, in fact, many of the subgroups of geckos are older than other lizard families, and also because of the number of species, their time was up. It was quite obvious they were going to be split into multiple families. And here on the left, you have a phylogeny. 7 families. Now, the trouble with that is you do get ecological categories. You get desert geckos, you get gliding geckos, you get rainforest leaf litter geckos, and you get each of those categories in more than one of the new families. So I'm giving you 3 examples here, 3 of the new families with at least 2 of the categories represented. And these are amazing examples of convergent evolution. The problem is, for the average person, it's no longer easy to recognize what a family represents. So where do we go next to come up with a manageable list of living organisms for public consumption? Perhaps this screenshot of what you get when you search the word animal may be the easiest answer. So the public is basically limited to focusing on the larger, morphologically distinct taxa, and to a great extent ignores the rest. This is a trait-based approach, as trait-based as they come. There are a couple of other flagship sort of approaches out there, and some of them are quite fun. The EDGE approach, which combines phylogenetic information and conservation status, is one of the more successful such approaches. Including conservation status here is, of course, useful in channeling funding in the right direction, but this means we're not talking about a natural classification per se. This is one proposal that caught my attention, and it's been around for some time. This is 1999 paper by Genovese and Glenn Johns. And what they're saying is, why don't we just take the phylogeny, and instead of using the classic higher taxa that we've used so far, let's use this phylogeny, a dated phylogeny, and slice it at different depths in time, and then use whatever comes out of that as higher taxa, or in place of higher taxa. Of course, there are limits. If you go deep enough into the past, you're going to land up into a mess of unicellular life that holds the deepest divergences, but they don't really mind exchanging genetic material with each other the way we exchange phone numbers. But if we just stick to the larger living beings that most people are likely to notice, maybe it's worth a shot. There are still difficulties. How do you persuade someone that an elephant and a mouse are the same, but there are 5 different types of salamanders? For the purpose of research, let's for now ignore this sort of concern. See how things pan out on a map or in a biotic assemblage. Perhaps there is some explanatory power in this approach that may be worth exploring. I found it particularly exciting that we stay phylogenetic, but we stay out of phylogenetic diversity. I was a great fan of phylogenetic diversity 15 years ago, but I increasingly feel it's a black box. It mixes old lineages, recent radiations, and everything in between in one number that really has very little meaning left at the end. What we're doing here is unpacking phylogenetic pattern, and we hope it will make it comprehensible to the human mind. So we define ancient lineages in vascular plants and in tetrapod vertebrates, because that's where we could get enough data, and started to look for global distribution patterns and for ecological patterns, and see how much we can do that is commonly done with species. So let's see what we got. So this first study that I'm going to present to you is the broadest in terms of taxonomic coverage, in the sense that we pull data for tetrapod vertebrates and for seed plants, and we map them globally using Olsen and colleagues' ecoregions as the basic geographic units. I'm going to put a slide like this at the beginning of each study just to indicate, did we look at plants, did we look at vertebrates, did we look at both, and were we studying things at the level of regions, that is on a biogeographical level, or were we studying things on an ecological level? So whatever you're seeing in red is what we actually looked at. Whatever you're seeing in black is the background, is stuff that we studied in other studies, but maybe not in the study that we refer to. So in this study, for example, we looked at vascular plants, with the exceptions of ferns. That's why I left one branch there in black, but it's of course it's a whole grade. It's very simplified trees that I'm presenting here, just to give you an idea what we did and what we didn't look at. And in this study, we looked at the biogeographic pattern, not at the ecological pattern, not at the types of environments. And we looked at two depths in time. We basically sliced the phylogenetic trees of vascular plants and tetrapods twice. We sliced it at 201 million years ago, and we sliced it again at 66 million years ago, because these are the times of the last two mass extinctions. So we placed the study in a hypothetical deductive context, in the sense of, let's look at the lineages that survived the last mass extinction, and then separately, let's look at the lineages that have survived the last two mass extinctions. Of course, it's not to say that the places where you find these lineages today are the places where they survived the last mass extinction, and even less so in the case of two mass extinctions ago, although this may be the case with some of the more range-restricted lineages. But there were clear cases where there were substantial diversifications and range expansions subsequently. So essentially, we took the phylogenetic trees of tetrapods and seed plants and intersected the branches with two lines, one at 66, one at 201. Any branch that got intersected at one of these points, that's one lineage for the analysis at that depth. All the species currently alive descended from that ancestor were pooled as one lineage, and all their distributions were pooled to represent the current distribution of all descendants of that one ancestor. So in terms of seed plants, at 201, about 1/3 are gymnosperms and the other two angiosperms. Tetrapods, amphibians are a big chunk of what you're seeing, and multiple lineages in all three extant lysamphibian groups. That's the Thessalians, the Urodellans, and Anurans. And then a handful of groups of reptiles, 3 mammals, whereas the birds and crocodiles are one lineage each. For 66 million years, we focused on specific subgroups in both cases, some that are overall globally more widespread, so as to partly replicate patterns for the whole group at 201, but look for differences in detail. The Campanulidae and the Poales for plants, with daisies and grasses being massive families, but in both cases we also have narrowly distributed groups. For tetrapods, we took the birds and the mammals. This is what we get. These are the maps Sid produced, and in all the other studies further down, Sid is basically the map man, So at 66 million years, which are the two planes on the right, we reconstruct for lineages what could be called a typical latitudinal pattern, the way we see it today in species richness. When looking at weighted endemism, so that's bottom right, we get a few interesting hotspots in the Andes, in the Cape, in Queensland rainforest. The really interesting part is when we go further back to 201 million years ago. So this is no longer a typical latitudinal pattern. We get high lineage diversity in the humid parts of the northern hemisphere, with hotspots in Eastern North America, along the Adriatic coast in Europe, Japan, South China, but also maybe more predictably in the Amazon and Southeast Asia, and also in New Zealand. These hotspots can be explained by the presence of often just one or two narrowly restricted ancient lineages. So in Europe, it's the Olm or Proteus. In New Zealand, is the Tuatara and Leopelma frogs. East Asia is mostly plant lineages. In a sense, it's a bimodal pattern, because on the one hand, you have large continental blocks with ample freshwater supply over the geological ages, where amphibian lineages were able to survive climatic changes. And on the other hand, we have highly isolated islands, where ancient lineages with relatively low competitive abilities are hanging in through the global rise of more competitive ones. And both biotic surges of new lineages and massive climatic changes were associated with the mass extinctions that we're actually looking at. Southeast Asia might be a combination of both of these patterns. So on the one hand, it's got continental features, but it also has insular features. And Southeast Asia is the one that's most consistently diverse across all four planes in this figure. So we took this Southeast Asia hint. I bet you didn't think this was going to be a Southeast Asia presentation, and most of it is not. We took the Southeast Asia hint. This is where most ancient lineages across the board are. Let's go there. Let's get some data and try to see what we can do by mapping ancient lineages, not just geographically, but also across ecological conditions. Now, Southeast Asia is not only good in terms of how many ancient lineages it has, it's also giving one plenty of options in terms of how to play with environmental variables. And what we found most exciting is that you can find vast areas of lowland rainforest with virtually identical climate, but a huge diversity of soils. So you can look at soils with minimal confounding effects from climate. So we did some fieldwork in Brunei on the island of Borneo, looking at plant representation across 9 forest types, and we were lucky to have a fantastic local team to help. 3 of the forest types were mangrove types. You get very diverse mangrove environments around there. And we had comments like, why do you mix things? Mangroves are mangroves. We had to say mangroves are plant assemblages living in very specific soil conditions. And that's exactly why we want them. Probably sounds like we only went there to have fun. There must be data out there to look at what we wanted to look at without actually doing any fieldwork. And you may be surprised. Most rainforest studies are trees only, or in some cases, epiphytes only, full plant lists for rainforest plots with soil data, and especially including ferns, because we wanted ferns here for ecological pattern. That sort of data is actually hard to come by. That being said, it certainly was fun too. That's just to say we did all plant, ferns included, no animals. We looked at the environments, but we also looked at biogeographical pattern towards the end of the study, and I'll get back to that with details. And the plant lineages we looked at were 120 million years old. I'm just quickly going to run you through some of the plant pictures from 3 of the forest types. This one is just your typical lowland rainforest with good soils and pretty much everything in terms of plant lineages. This one is secondary forest with slightly more light at ground level and a whole variety of ferns, and amongst the gymnosperms, Gnetum, which represents its own ancient plant lineage. And this one is Pizzwam Forest with one dominant dipterocarp species and an understory of gingers and pitcher plants. And by the way, pitcher plants happen to be caryophyllads, which are dominant globally in arid environments, so that's a different one for them. So for the sake of time, I'm not going to take you through all the forest types. I'm just giving you here a summary of the 30 120 million year old lineages that we had in at least one plot. So I'm only giving you here one line drawing for each lineage, but more often than not, there were several species in each lineage. So this is the summary of how many species in each lineage were present in each forest type and how dominant they were. For each lineage, you see the stacked boxes equal in number to the species present there, and the size of the boxes is proportional to the log transformed cover value for each species. You can see that 3 of the lineages are dominant in pretty much all the forest types, and all of these are eudicots, and they're closely related to one another. They're the Malvidae, Asteridae, and Fabidae. This is even the case in mangrove forests. So we thought, wow, we wonder whether this is the case globally. Of course, we didn't have cover values globally, but we have species richness values and we have geographic range. So this is what we got. The size of the circles here is soil niche breadth at our Bornean plot, and the 2 axes are global range and global species richness. I'm not going to bore you with all the p-values, but trust me, one can predict global success in these lineages by simply knowing the variety of soils one lineage can live on in one region. Of course, the soil niche breadth was not necessarily present in the common ancestor 120 million years ago, but was often achieved subsequently in a descendant that then used this acquired ability to colonize much of the world, including our sampling site. That being said, in some cases, it may have been there already. This would explain why 3 lineages that are closely related are doing well globally, and the ones distantly related to them are doing poorly at our sites and elsewhere. The ability may have had to do with other traits, though not soil related, perhaps reproductive biology, wood anatomy, and so on. The short of it is, though, one can use ancient lineages to study ecological niche as well. So we decided, let's see what else can we do with lineages that people usually do with species. So Erwin ran a canoco initially at the species level with our Bornean data. It looks like a pretty regular canoco, 56% of the variation explained, which is good. And you can actually guess the clusters that represent different forest types, each with their own characteristic species. This is a second canoco using plant families. At this stage, forest types are starting to be mixed, but the explanatory power is actually higher. And then here's a third canoco for the 120 million year old lineages. There's no real structure, but most of the explanatory power is retained. This is basically the sort of stuff Andreas Printing and his collaborators were saying in 2001 about phylogenetic conservatism across taxonomic ranks, except we have more lineages at a deeper level in the tree, because we chose a study region that has plenty. So we can basically probe how deep phylogenetic conservatism goes. So one can use ancient lineages in canocos too. Getting back to the global distribution data for these lineages, we played around to see whether we could use them in a global regionalization exercise. At 120 million years, as examined in the Borneo study, we still had too few lineages to get a robust analysis to this effect. So we moved the cutoff to 100 million years, and some of the lineages were split a bit further, and explanatory power got better. We left out ferns and allies because the 100 million year lineages in those groups are mostly cosmopolitan, so they add little explanatory power. We started by mapping lineage, diversity, and endemism. You may remember that in the first study, at 66 million years ago, we did not look at all plants. We focused on campanulids and poalids. So this is in fact the first set of maps of this kind for lineages in that age range. So same as in previous studies, you can see highest diversities in East and Southeast Asia, and also in Central America. The most interesting part are the similarity patterns. So Sid did two analyses here, a parsimony analysis of endemicity and the basic UPGMA clustering procedure. They gave us practically the same. There are three major clusters, basically identical to the three global realms which were proposed by Rappaport. An Antarctic realm, a holotropical realm, and a holarctic realm. This is actually the first analysis of this kind to recover these three major clusters globally. Keep in mind our Antarctic realm is fairly weak. What you're seeing here on the right is the PAE. On the UPGMA, you can see that New Zealand and temperate South America connect quite close to the root. So it's not a very strong cluster, but nevertheless, these two regions are closer to each other than they are to anything else. What is the stronger pattern here is that there's a major divide between tropical and northern temperate floral assemblages. It had already been shown that this is the case with plants more than vertebrates because they are by and large better dispersed. So once again, this is not some preserved Gondwanan Laurasian pattern. It's younger lineages within our old lineages moving around within their climatic niche. Sometimes several families in each old lineage moved all over the place, but in doing so, they lend even more explanatory power to older lineages. So we can use ancient lineages in regionalization too. The last study I will present is not about ancient lineages, it's an alternative approach. We published this in 2013 and we're still trying to get a follow-up paper. So here we looked at what we defined as cosmopolitan lineages. We basically defined the minimum spanning tetrapod lineages that can be found over much of the world. If that's one species, that's good enough, that's a cosmopolitan lineage. If it takes 1,000 species to make something we can call cosmopolitan, so be it. That's as narrow as cosmopolitan lineages get in that group. These are just examples of things we call cosmopolitan. They have to be in at least 60% of world's ecoregions. So they basically have to be present in most, or preferably all, of Eurasia and Africa, and then at least parts of the Americas. They may or may not be present in Australia, and many of them are not. These are the 83 lineages that we got that meet these criteria and the relationships between them. As you can see, lots of birds, mostly non-passerines, a few mammals and squamates. Chelonians are one lineage, and there are two frog lineages. These line drawings are basically ones that people around the world should be able to recognize and name. In English, most of them have single word names, and that in itself is quite telling. Mapping diversity of cosmopolitan lineages may sound like a bit of a non-task, but it's still interesting. Most interesting, in fact, is where they are not. As you may expect, this is mostly in New Zealand, Australia, New Guinea, Madagascar. That's where cosmopolitan lineages are missing. Just for curiosity's sake, I will add that most of them are in Southeast Asia yet again. In fact, there are two ecoregions in Southeast Asia where all 83 lineages are present. This is not the case anywhere else in the world. What we also thought would be interesting was to find out where they may originate. So we looked where the sister lineage of each of these lineages is found. In a few cases, two cosmopolitan lineages are sister to each other, which is often uninformative, but in other cases, the sister lineages are narrowly distributed. And most of these are once again in Southeast Asia, but also some of them are in the savanna-dominated parts of Africa. These are the sister lineages we listed as relevant in the paper, the ones you see in the pictures. The phylogenies have changed a fair bit since, but they've changed in both directions. In some cases, the sister lineages are no longer the ones from these regions, but in other cases, the new sister lineages are. So the overall map that you see here tends to stay the same. There's also the small matter of how a lineage becomes cosmopolitan. And there's very little doubt that it's by being dispersed well. Here's a massively significant comparison between how many species it takes for a lineage to be cosmopolitan if they can fly versus if they cannot fly. In fact, out of the 83 lineages, it's only 1 or 2 here where continental level vicariance may have been of some relevance. Here is one of those, and it's the slowest moving of the lot, the chelonians. The root divergence in chelonians is between the two major groups, the Cryptodira and the Pleurodira. The former is mostly Laurasian, the latter is mostly Gondwanan. Cryptodira on its own narrowly misses the cosmopolitan definition cutoff, so we have to include Pleurodira to make it a cosmopolitan lineage. But basically the message we get out of the story is that if you're doing phylogenetics, continental vicariance is a hypothesis you can safely ignore in the vast majority of the groups you might be studying. In fact, like I said, we want to produce a follow-up study with divergence dates to finally settle the matter of these being all about dispersal and even put some distances on those dispersal events. If any of you in the audience has a bit of time on their hands to crack this one, please give us a shot. So this is all we wanted to tell you about our studies, but we're quite excited to see that there are a couple of recent papers unrelated to our work doing similar stuff, essentially using lineages to map distributions and to study ecological pattern. Some call them clades. We prefer lineages because saying clades implies that one holds on to certain assumptions, but in this context, it's basically the same thing. So to end with, I would like to go back to the Borneo study because it's perhaps the bit of the presentation that best reflects the title, the one that's most suitable for a narrative. You take 30 plants alive 120 million years ago and follow them through the ages. Some spread and diversify massively, others remain localized and perhaps diversify a bit. It's what Janssen and Dionysius called, in a different context, the fate of clades. I think we have shown that we can actually illustrate many of the basic patterns in ecology and biogeography using just a handful of lineages that even a layman could easily grasp and recognize. Much of the work is exploratory, and there may be better ways of doing this. Our message is that biodiversity can be simplified, both for research and for the general public, and at the same time, one can discover new and exciting patterns. Thank you very much to all those who helped and to those who provided funding. And thank you guys for your time. We're looking forward to hear your questions.